Hello everyone, welcome to Tamali's classes. Today's topic is complement system. This system is very important as it helps our body fight the infections and also clear all the damaged cells. Okay, so this process will help in attacking the pathogens by attaching with their cell membrane and then killing them and also removing them from the body, and also it will clear the damaged cells that are present in the body. It will enhance some factors, some defense mechanisms. Also it will create inflammation in the body and help in the immune responses. Okay, so let's start the topic today. So first of all, we'll talk about the complement system and what is it and who discovered it. So first of all, let's say complement system is what? It is a system of plasma proteins. There are many proteins involved in the system which are normally in an inactive state, but they get activated when they recognize the microbes or when they recognize the pathogenic microbes. Okay, so not only microbes, whenever they recognize any pathogens in the body, they will get activated, and then after that there will be a cascade of reactions. So one protein will lead to activation of the other protein, and this way it will continue. There's a cascade of reactions happening in this body. So the surface of the pathogens, which will help to kill them by destroying their cell membrane. Okay, so this was given by Jules Bordet. This system was given by Jules Bordet. So what is the principle basically of complement system? So complement system, it's a series of more than 20 proteins that I've already told you. It's staying in an inactive state in the blood and in the tissue fluids. So when they recognize any components of any microorganisms that are pathogenic in nature, they will go and attach to it, and they will create some reactions on the surface of the pathogen, helping the the cell to kill them. Okay, helping the killing of those pathogens. So there are many proteins out here involved. We'll get to know that in my next slides. So basically, they are one of the most important protein is out here, C3 proteins. So C3 protein, what happens when this C3 protein gets activated, and when it gets when it gets digested, when it gets broken down, or you can say it gets cleaved. Okay, it gets cleaved into two other components like C3b and C3a. So one of them out here, C3b, is opsonine, which acts in this defense process by promoting inflammation. Okay, also the other one is the C5. So C5 is also out there very helpful because it will attract the macrophages. I told you that there are some factors which will be present in the cell which will be responsible for immune responses. So one of them is macrophages, neutrophils, and also mast cells. So these are all those components, all those factors, all those components actually in the immune system, which will further help in further help in defense process. They will further help in fighting the infection. Okay, so they will help in the killing of the microbes, the pathogenic microbes. So we'll get to know in details about it. So there are three pathways of complement system: the classic pathway, the mannan binding lectin pathway, and also the alternative pathway. So these three pathways are it can happen, which will lead to the fighting the infection and killing of these microbes. Okay, and also removing the damaged cells from the body. So let's talk about the pathways. So first, I've placed out the three pathways together. You can see in your slide: classic pathway, alternative pathway, and mannose binding lectin pathway. So you can see in the classic pathway out here, so there's an antigen antibody complex formation out here. So C1 is another protein. So C1 protein out here, it gets cleaved into C1q. There are many forms, okay, of C1 protein. So C1q, C1r, and C1s. So just look at here. I'll show you. See here, the C1 out here. Okay, C1 is out here, C1q, C1r, and C1s. So this C1s, what does it do? It will go and bind, it will activate this C4, leading to cleaving of the C4 into two other components, C4a and C4b. So again, this C4b will go and bind to this C2, okay, and this C2 will again be activated and it will cleave into C2a and C2b. So C4b will bind to the C2b component out here, and it will form C4b2a. Okay, so this is formed out here, and then this will help in the along with this. So, and C3 convertase is that enzyme which will help in the cleaving of C3 into two components, C3a and C3b. Okay, so similarly, let's talk about, we'll get to the lower part below of this after the C3 step. So let's talk about the initial steps out here. In the alternative pathway, if you see out here, the activating surfaces, the protein will go and bind out there, and there are some factors involved, factor DH1. Okay, so they will again activate the factor B out here, and again they will form this C3b, capital B, small b complex. So out again factor B will have two components out here, and with the factor capital B, small b component, C3b will bind to form this C3 convertase part. Okay. Again, if you look at the mannose binding lectin pathway, this is different from the classical pathway. Only in certain thing that in classical pathway, the antigen is binding with the antibody. Okay, so by looking at the pathogenic surface is involved out here in the classical pathway, but in mannose binding lectin pathway, we are talking about the glucose or mannose present in the microbes, in bacteria and microbes. So what's happening out here is that MBL, mannose binding lectin, what they will do, they will recognize the mannose and the glucose on the surface or present in the bacteria or other microbes in the surfaces of the pathogen. So they will identify those, they will bind to those, and it will also lead to activation of MASP1 and MASP2, which is serine protease. Okay, mannose binding lectin associated serine protease 1 and serine protease 2. So this will go and activate this MASP1, MASP2 part, and they will activate the C4 and C2 part. Okay, so they will activate C4 and C2 part, and again it will lead to formation of the similar like classical pathway. It will be forming C4b 2A component. Okay, this complex will be formed out here. So you can see this three complex, and they're all coming to the same step, C3 convertase step. Okay, so just to mention you for your exam, it will be helpful. This MAP44, this acts as an inhibitor. Okay, this will inhibit this MASP1 and MASP2. So since they will inhibit this, so there will be no activation of C4 and C2 proteins, and this pathway will be restricted. Okay, remember this MAP44 is an inhibitor which will inhibit MASP1 and MASP2 functioning. Okay, so now coming back to the C3 convertase part out here, so you can see all these three pathways, they are leading to the same step of C3 convertase path. Okay, so out here what will happen, the C3 convertase will cleave down C3 into two components, that is C3A and C3B. So if you see out here, C3A is activating the mast cells and releases histamine, and C3B will attach to the pathogen and opsonize. Okay, so this is very important out here, C3B part. So this C3B part will again activate this along with the C5 convertase will activate this C5, because again C5 convertase enzyme will cleave down C5 to many components. Okay, one is C5A and one is from C5B to C9. Okay, so this C5 will attach with will bind the target cell, which is being targeted by the immune system. So the C5 will bind the target cell, and it will be cleaved into C5A and C5B to C9. Okay, there's an entire part, C5B, C6, C7, C8, C9. So this is an entire part out here in one way, and this is another. So C5A is out here. What is the functioning of C5A? It's helping in activating of mast cells, has a chemotactic response, and also an anaphylaxitoxin. Okay, anaphylaxitoxin functioning. Okay, so this is gone. Now we'll come to the C5B to C9 complex. This is important because this is known as the lytic pathway. Okay, this is very, very important. This is known as the lytic pathway because this is also known as the membrane attack complex, MAC. What is this membrane attack complex? So the name itself says that it attacks the membrane. So this complex will attack the membrane. It will get inserted into the bacterial surface, and it will break down this cell membrane, damage it, and it will lead to the death of that microbe by lysis. Okay, lysis means to break down. So this is how they will kill those pathogenic bacteria. Okay, now how does complement system protect against infection? Number one is that it will generate large numbers of activated complement proteins. Okay, so it will generate large number of activated complement proteins. As you have seen previously that they are inactive, they are in inactive state, so they has to be activated. So first they will activate that, okay, proteins, and then this will go and bind with the pathogens. They will opsonize them and they will engulf them, okay, by using phagocytes. Okay, number two is that the small fragments of this component proteins, they will act as chemoattractants. Okay, we have seen out here, this is the chemotactic pathway. So they will act as chemoattractants, and they will recruit more phagocytes on that site where this complement system is going on. Okay, where this activation process is going on, they will attack more. They will recruit more phagocytes, and they will attack the surface, and they will also help in activation of these phagocytes. so that it can further function. Number three is that the terminal components. Okay, terminal means that in the end you can see those MAC, membrane attack complex. What they will do, they will damage the bacteria by creating pores in the membrane. See, the membrane is out here. As we have seen that they will insert into the bacterial membrane. How will they insert? By creating these pores. So they will insert, they will kill the cell membrane, and that is how they will attack the microbes. Now, suppose there's a deficiency in the complement system. In complement system, what can be the effects of such deficiencies? So number one, if we see out here, the C3 and factor B. So this is very important. We have seen this C3 part out here. C3 is very important so that there is a cleaving of C3A and C3B. So that will be affected out here. Factor B we had seen in the alternative pathway. So if there is a deficiency in these complement components, then it can lead to several bacterial infections. Okay, then these can be very severe. Also C3B we have seen in the alternative pathway, and then C6 and C8, that's in the membrane attack complex we have seen. So they can lead to severe dysentery infections. Now, if there is a deficiency in the C1, C4, and C2, these are the early components that were used in the beginning of the pathways. So this can lead to this SLE, systemic lupus erythematosus disease, and also glomerulonephritis and your polymyositis. Okay. Last is the C1 inhibitor. So suppose there's an C1 inhibitor, if it's present, and it will lead to some hereditary angioedema. Okay, so these are some of the diseases caused because of deficiency of the components in complement system. So thank you so much. I hope you have liked the class. Do share it with your friends, and also do subscribe my channel for more such videos. And thank you so much.